Hello and welcome to TechMed 36. Today we're going to be talking about transcription in eukaryotes. So things that we need to understand before we begin are going to be the difference between what are introns versus exons. So this is an important subject that we need to understand first before we can begin, because later on when we talk about the splicing and how we actually attain mRNA completely, well, we need to understand what the difference between an intron and an exon is. So, if I was to kind of draw like a little strand of DNA, I'm just gonna put a big white line right here. And I'm just gonna add some color and I'll put some red right here on this side. Maybe some blue. Orange, some ugly green, and let's say pink right here. Okay, so now that we have this here, we can start kind of to begin with what these colored sequences mean. And the only thing that we need to understand from this is that introns are going to be intervening sequences that are just going to kind of stay in the nucleus but are not going to be expressed. Why? Because they're just staying in the nucleus and we're going to try to get rid of them at the end. While exons are quite different, because these, which are the exons, well, you can put all of them, the ones that I just colored, are the ones that are going to exit the nucleus and are going to be expressed. So this all practically occurs during, after transcription, after this has become mRNA. And we want to turn it, this is actually going to be hnRNA, and then it'll become mRNA. So here I'm going to put H and mRNA. And we want this to turn into, practically mRNA. And how is that going to happen? Well, now after going after what is going to be called splicing, which I will explain later on, kind of like by the end of the video, after we have talked about transcription, this is just so you guys can understand something general. Well, we are going to have this right here, which is going to be our final mRNA sequence, which does not include the introns, because this is now going to exit the nucleus. And it's going to go to a ribosome, and it's going to go later on through translation, so it can become, translated into a protein. So, now that we kind of understand this, we know also that introns are larger than the exons. And just so we have this general idea for now, we can now move on to what transcription is. So, transcription is going to be divided in 3 steps. And our first one is going to begin with initiation. 2 is going to be elongation. And 3, which is the final one, why? Because there's 3 steps, is going to be termination. So, now that we understand about these 3 steps, now we can also talk about, right now, these are just kind of like our little outlines. We can talk about another thing that we need to clarify, which are the enzymes that are going to take place, which are going to be RNA polymerases. If you guys already saw the replication video, we were talking about DNA polymerases that were just replicating the DNA. But now, because we are creating RNA out of the sequence of DNA that we had, we're going to need RNA polymerase. And when we talk about eukaryotes, we're going to have 3 kinds, which is going to be, of course, 1, 2, and 3. So here, there's no really big theory to what it is. It's just 1, 2, and 3 in Roman numerals. And each of these is going to do something different. It's going to codify different kinds of DNA. So, number 1, we are going to be making what is going to be known as rRNA, but these are going to be large. And rRNA is just ribosomal RNA. Number 2 is going to create what's going to be known as pre-mRNA, which is also known as what we just mentioned, HN-mRNA. It's also going to create snRNA, snORNA, and miRNA. And 3 is going to create tRNA, which we also be talking about later on when we usually talk more about translation. It's also going to create rRNA, but these are going to be small in comparison to RNA polymerase 1. And it is also going to make some miRNA. So now that we understand the 3 kinds of polymerase that we have, usually the one that we will be talking about transcription is going to be RNA polymerase 2. And now what we're going to be talking about is the DNA sequence and what is going to be occurring and what we need to actually focus on. So what we're going to be focusing on is special regions inside or in the DNA that are extremely important for us to understand transcription. So what are these areas? Well, first we're going to be starting with drawing a nice DNA strand. So I'm going to go create this right here. And in kind of like grayish, I will make the other part, because remember that DNA is a double strand. Okay, so here we're going to have this one ending in 3', this one's in 5', 3' right here, and 5' right here. So we're going to have different regions in the DNA that are going to help us begin the transcription. So first, I'm going to make one right here, and I'm going to be indicating that this right here is going to be a certain piece out of a really important part, which is going to be all of this right here. And it's going to be known as the core promoter. The core promoter is the one that's going to contain what's known as the promoter sequence that I actually put here in blue. So I'll put an arrow right there. This is the promoter sequence. And what's so important about the promoter sequence is that this right here is going to be rich in adenine and thymine. And usually we're going to know it as either the Tata box or the cat box. If you notice, these are kind of rich in adenine and thymine. The Tata usually can have 2 A's at the very end, but I mean, it's just so we can understand that these are the boxes of how we know them. So this right here is going to be known as the Tata box. So I'll even write some letters right here. I'll put TA, TA. So this is the Tata box. And the important thing about the Tata box is that this is where, practically the transcription is going to initiate, or it's going to be recognized for initiation. And we're going to find this in negative 25 nucleotides from the transcription initiator element. In other words, where the transcription will actually begin. So we can actually put that right here. I will put it in blue and brown. Let's put it in brown. So let's say it's right here. And this one is going to be +1. And this area right here is just known as the initiator element, and it's where the transcription is going to begin in this +1 area. So I'll even put an arrow here. That way we know what it is. And it's the initiator element. The initiator element. Okay, so now that we understand this, we'll also know that in all of this promoter region, we're also going to have something known as a regulatory promoter. and I will put that in green. Let's say it's this area right here. And that is the regulatory promoter. And this regulatory promoter is going to be upstream from the promoter sequence. Upstream meaning that it's actually going to be going negative. This would be downstream. So let's say if we were just to kind of switch it this way, you know, just like a big side, this is going upstream and downstream. So if we go upstream, we're going to find this regulatory promoter. And this is where certain transcription activating proteins are going to be affecting the transcription velocity. So in other words, this is where we can either be making it slower or making it faster. And also, we are going to be finding another area which is going to be, it can be very far away from our beginning genes here, which are going to be known as enhancers. So I'll put here enhancers. And what are these enhancers? The enhancers are going to be some stretches of DNA that are practically going to be altering the gene expression. And what's going to occur is that, let's say I have a strand of DNA again. And I'm going to put here in pink our enhancer area. So right here we have an enhancer. So what's going to happen is that certain transcription factors are going to come here. So let's put TF for transcription factors, are going to go right here, and they're going to practically bind to the enhancer. And what's going to happen is that, let's put our data box again. Let's say it's all the way over here. What's going to happen is that now it's going to form a loop. So we're going to have a loop form. In other words, this is going to come practically like this. So the transcription factors that are right here are going to be able to interact with the machinery that is going to be in the core promoter. Because later on, let's say that now this guy has had its transcription factors, and they're right here binded. So when the loop is formed, instead of seeing the DNA like this now, this is practically how it's going to look like now. It's going to loop a little. Which means that now, where we had our data box right here, and its transcription factor, well, now we're going to see that this enhancer area is going to be way closer because of this loop, and the transcription factor that the enhancer had is going to be able to interact right here with the transcription factors that are in this core promoter. And this right here is going to alter practically how the gene expression is going to occur. So this is practically what occurs with the enhancers and what it's important for. Of course, we will also have sites that are going to be known as silencers. So I can even put here versus some silencers, which are also enhancer regions, but instead of them practically promoting or enhancing gene expression, well, these are going to be repressors. So in other words, they are going to practically be inhibiting gene expression. So now that we have these basic concepts down, we can actually begin with the 3 steps that we have mentioned right here about initiation, elongation, and termination. So first we will start with, of course, what's going to be initiation. So initiation. So what occurs during initiation? Well, this is going to begin with the forming of what's known as our transcription apparatus. So apparatus. And our transcription apparatus is going to consist of various things. One thing being our RNA polymerase. So I'm going to put RNA pol, just put as pol. And it's going to be here especially too because we will be talking about us making mRNA. So we need RNA polymerase too. We're going to need some transcription factors. And these we're going to have actually various. ones. So we're going to have TF 2A. Why? Because transcription factor for polymerase 2. So this is where that 2 comes from. We're going to need TF 2B. We're going to need TF to, can you guess it? D. No, it's not C. Ha. Tricked you all. Okay, so then we have TF to F and TF to, can you guess it? H. Yeah, it's not G. Okay, so now that we have these different transcription factors, then we're also going to need something known as a mediator. And this right here is going to be our transcription apparatus that is going to be necessary for the initiation. So our first step, so I'm going to go all the way up here and right there. So our first step in this part of initiation, so I'm going to put initiation here again. So our first part of here of initiation is going to be the formation of, well, our union to the data box. So if I was to draw this strand of DNA, and we have our data box again, so let's put that in blue again. This is our data box. We're going to have a union with transcription factor 2D. And this transcription factor 2D is going to create or contain a subunit right here that we will know as TBP. And this TBP is the one that is going to recognize the data box. And then once it has recognized it, it's going to practically tell other transcription factors to come and join the party. So the TBP is just going to go and recognize the data box. Once it has recognized it, it's going to practically tell the other transcription factors to come and join the party. So here we go. We'll have this right here. Our data box. And now this joining of TFD, so TF2D, with the TBP that had recognized, well, the strand here, or that data box. And now it's going to be telling all its friends, hey guys, come join the water, you know, it's all fine. So here come the rest, which is going to be, well, the TF, 2A, then the B, then F, and H, and also our mediator. So all of these guys are just going to come and join the party. So now, once they have joined, these guys right here, so let's say I'm going to move down right here. So now that these guys have just joined this party, so we're going to have this right here that I've just joined. They're also going to bring another person, which is RNA polymerase 2. So this RNA polymerase 2 is now going to join, which I will put here as ugly green. And it's practically going to come right here. And it's practically going to begin everything, you know. But what else needs to occur? Well, we're going to have a region here that we had said, which is going to be our regulatory promoter. This regulatory promoter is going to have its own transcription factors, which are the transcriptional activator proteins. So transcriptional activator proteins, or TAP, that are going to come and join right here. And these, by certain coactivators, are going to help and stimulate for transcription to occur. And once that also happens, we're going to have enhancers. So we're going to kind of move it down just a little. Because now what's going to happen to this strand of DNA is that it's going to go and move like this. Why? Because we had that data box region, which was right here. We're going to have our regulatory promoter right here. And right here we had what is called our enhancer, if you guys remember how we were talking about it. So now this enhancer is going to have its own transcription factors that are going to come and practically tell them, hey, let's join the party. So here I'm just going to put this RNA polymerase. I'm not going to put the rest of the transcription factors because it'll just take a little too long. But we're also going to, this is also going to have, the enhancer is going to have its own taps come in. In other words, our transcriptional activator proteins that are going to come right here, and it's going to enhance, as its name says, enhancer. So once we have this, we have now practically have initiated by creating this transcriptional apparatus. So now that we have all of this, we can move on to the next part, which is practically after initiation, which is now elongation. And now what does elongation do? Well, here we're going to be actually focusing on the polymerase. So if I was to kind of draw the polymerase, I'm going to do this kind of like subunit here. I'm going to create another subunit here. Kind of join them a little. Just going to go like this. Create a little opening there maybe. And kind of like that. So here we have our RNA polymerase. So I'll even put there RNA polymerase 2. And now what is going to occur? Well, the DNA that has just come in, so I'm going to kind of put one strand here. It's going to kind of go up here. And in blue, I will create the other part of the strand. So it's going to kind of go like this. So what is polymerase going to do? Well, first it needs to uncoil the DNA so it can create the RNA. And practically we're going to be working on putting some pieces on the 3' ends. That way we can be able to practically elongate by creating the mRNA. So to do this, we're going to be seeing, as I will put here in, let's put pink, why not? We're going to have this being the exit and this kind of being like a little funnel where the new synthesized mRNA is going to be. So here we're going to kind of create this right here. I'm going to go kind of like that and then like this. And why? Because in this part we have the active portion of the enzyme, because once it has opened, what is it going to be doing? It's going to be creating these new base pairs. So it can be creating our mRNA, well, RNA technically, because this is going to be our pre-mRNA, and pink, because once it has come in, we're going to see this part being the 3' end and this being the 5' end of the RNA, mRNA, or pre-mRNA, that has now become elongated by practically just transcribing, or, well, yeah, it's going to be transcribing the DNA. So once that has occurred, we can now go into termination. So here now we have this strand here that has just been synthesized of mRNA, or pre-mRNA, because we still need to kind of edit it. So once we have just gone through this elongation, we're now going to go into termination. Now here I was just practically talking about RNA polymerase 2. However, it's practically the same concept when we talk about the other two polymerases. But termination is something that's going to be quite different, because we're going to have very different approaches when we talk about polymerase 1, 2, and 3. So 2 and then 3 over here. So the only thing I can mention about polymerase 1 is that for it to finish, it's going to require termination proteins. And what this means is that these termination proteins are going to bind to the DNA. And this is practically going to stop our polymerase. So now we're going to put kind of like a little stop sign here. It's going to say stop. And it's going to stop our polymerase. But what about polymerase 2? Well, polymerase 2 is going to be a little different because it's going to require of something called RAT1. And I will go into more detail on RAT1 a little later, because this is the one that is most studied and well known. So I'm going to be able to explain this one in more detail. And then we have R3, which is going to have a termination sequence in the DNA. And this termination sequence is going to be transcribed, which is going to be rich in thymine. And it's going to create a poly-U in the RNA that will terminate the whole transcription. So, now that we have established here termination, we are going to go into termination for polymerase 2. So termination, and this is polymerase 2. First, we're going to here draw our little strand of DNA. I'm going to do this. It's going to kind of open up like that. It's going to be a little close right here. Right here. Boom, 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 boom. Because this is where practically the whole, let's say this is where the sequence is occurring. I'm going to put here in this kind of like array. Just going to circle this around because this is going to be our polymerase 2. And right here, let's say, is our pre-mRNA. So here is our pre-mRNA. Just kind of goes right there like that. So this right here is our pre-mRNA. And we need to create our termination. So to first do this, our pre-mRNA is going to have an end site. And this end site is practically going to be sliced. So I'm going to kind of write like a little site here. Because that is where, or here I'll put it in ugly green. There we go. We have an end site. And in this end site, what is going to occur? Well, we need to practically splice it. So I'm going to put here that this is our 5' end. This is our 3' end. And this is going to be sliced, so it's going to be practically erased. So let's say I now do this, and boom. This is where it has now been separated, so now I can establish that this is now the new 3' end for this one. So I'm going to put 3' end here, and the 5' end for this one. So because this has occurred, now we can have what is going to be RAT1 kind of come here and start degrading it. So here, we're going to have RAT1 come in. And what's RAT1 going to be doing? It's going to start moving this way, kind of like degrading the DNA, or this mRNA, until it practically reaches the polymerase. So let's say it's right here, and it's going to start moving this way. So as soon as it starts moving that way, what's going to happen? So we're going to draw this again. being right here. So now rat1 has practically gotten rid of everything that was in its path, and now it's going to meet the polymerase. So there's been contact. So now that there's been contact, what is this going to do? It's going to kind of tell the polymerase, hey, you know, this party's bunk, let's get out of here. So this right here is practically going to detach. So here now it detaches. And these both kind of like leave. So let's say we go like that. And what's going to end up is just this right here. The DNA is going to go back together. And our little party goers are just left the building. And our little party goers had just left the building. Boom. Goodbye. And this right here is practically our whole transcription. But what have we just made actually? Well, all we have actually made, which is this strand that is right here, is our pre-mRNA. So it's still not done. Because what do we need to do? Well, we need to make this into mRNA. And this is going to go through certain steps of editing that have either occurred at the beginning of transcription, at the end of transcription, or, well, practically during. So when we talk about this editing, so I'm going to put editing now. We need to kind of remember what we were talking about introns and exons. So first we're going to be talking about our first editing that is going to occur at the beginning. So I'm going to put here at the beginning of transcription. And this one is what we'll be knowing as 5' capping. And what is 5' capping? Well, the important thing we need to know is that it's used to stabilize the mRNA. And it's going to protect it from the nucleus. It's going to protect it from nucleases. I apologize, not in the nucleus. And it's going to facilitate intron removal. So this is at the beginning of transcription. And how does this occur? Well, we can just kind of say it in 3 easy steps, really. So I'm just going to put here 3 steps. So now, the first step that is going to occur for this 5' capping is practically just the 5' end, as we were saying, is going to be added, this 5' end is practically just adding some nucleotides of methylguanosine. So now, how does this happen? Well, the first thing that we're going to do is that we need to understand that there are going to be 3 phosphates present at the 5' end. So let's say this is the 5' end. And what are we going to have here? Bonded to it. We're going to have 3 phosphates. Why? Because we are at the deoxyribose phosphate end, not the hydroxyl end that's known as the 3'. So what we need to do is that we need to eliminate one of these. So let's say we're going to eliminate the last one. So now we're going to be having this 5' end here. And instead of it having 3 phosphates, boom, boom, it has only 2. So now here it's kind of like, oh, where's my friend? You know? So it's going to have a little tear because he just lost one of his best friends. It's like, oh no. And he's going to need a new friend. So what's going to happen, we're going to have what's known as a guanosylation. In other words, that aguanosine is going to be brought in. So aguanosine, let me write that down. Guanosine is going to come right here. And I will put an image practically of the way it's actually supposed to look. This is just kind of like a big simplification. So guanosine is going to come in. And after guanosine has come here, and it's going to go with a bond known as 5' triphosphate bond. So a 5' triphosphate bond. And of course, this is known as guanosylation. So once this has occurred, what's next? Well, now we need to add a methyl group. So now who comes in? A methyl group. So this methyl group is going to come in by some methyl transferases. So let's put methyl transferases, which is the name of the enzyme that is going to help this methyl group come in. And it's going to add itself with this guanosine. So here we had that 5' end. We had our little phosphates there that had just lost their friend, but they got a new one. And here I will put in green what this guanosine end looks like. And the methyl group, which is going to be right here, as red. So this has just been added right here as the new friends or the new kids on the block. And this is practically known as, well, a methyl guanosine group. And that is practically how this 5' capping goes. And like I said, this is just so it can stabilize the mRNA, protect it from the nucleases, and facilitate intron removal. So this is just one of the editing parts, because now we're going to have another one. And this one is going to be known as polyadenylation. So I'm going to put polyadenylation. And this one is practically going to be an addition of about 50 to 250 nucleotides of adenine to the 3' end. So this is going to be adding, so plus 50 to 250 nucleotides to our 3' end. And this is going to occur after transcription, so we can understand that after transcription. In case you guys kind of get confused which one is before or at the beginning and which one's after, well, think of this polyadenylation. So we're adding a lot of A's, a lot of adenines. So A after transcription. And this is so we can have major stability to the mRNA. And also it's going to help with our union. So this is going to facilitate union to the ribosome. It's going to facilitate its union to ribosome. And practically the only thing that goes is that now that we have this pre-mRNA, so let's say we're going to put our pre-mRNA, which is right here. Hello. So our pre-mRNA is right there. And what's going to happen is that there's going to be a certain sequence known as a consensus sequence. Let's put that right here. So this area right here is going to be known as a consensus sequence. And this consensus sequence is going to be recognized, which is usually going to have well, let's say the sequence is going to be AAUAA. Some books, that's how they usually write them. So AAUAA. And we are going to have a splice. And that's usually going to occur about 11 to 30 nucleotides after. So let's say right here in green, I'm going to go right here, and I will indicate that right here are 30 nucleotides. Not always 30, it can usually range from 11 to 30 nucleotides. So after that, we're going to have a region which is going to be known as the cleavage site, right here, after those. So now we have the cleavage site. And the cleavage site, like it says, is where it's going to be cleaved. And after that cleavage site occurs, we're going to be adding this tail, a poly A tail. So now our new pre-mRNA, let's say, it looks like this. We had our 11 to 30 nucleotides, which are right here in green. It has just been cleaved. So if you notice, it looks a little shorter. We don't see this rest of the place here. Now it's just right there. And what's going to happen? Well, we're going to have the addition of some adenine nucleotides. So let's put that in red. So these poly A tails, so if I was just going to go like this right here is just going to have a pure adenine. That's why it's just known as poly A tail, because there's just a bunch of adenines right there. And of course, in case you guys had forgotten, this is at the 3' end. This right here was the 3' end. Oh, that's not supposed to say 3. Let's get rid of that because that's supposed to say 5. So here we're going to put 5' end. Okay, so now that this poly A has added, well, now we can actually have this, stabilization and facilitation for the union of ribosome. So these are two edits right here that are really important, but not, well, I mean, they're all equally important. I can't say one is more important than the other because they all are kind of necessary. But now we're going to go to another one that is a little longer in understanding what happens. And this is going to be using our knowledge that we had said over here about introns and exons. So I'm going to go into another area. So here we're going to go into a little longer process. There's going to be our third kind of editing. And this will be known as splicing. And this splicing is going to take place so we can actually get rid of those sequences or these introns. And now, here what we need to understand, so here I'm going to draw this area right here, and I'm going to put that as exon 1. Exon 1. There we go. Exon 1. A little space here. Then I'm going to go over here and I'm going to put exon 2. Why? Because we're going to have an intron in the middle. And we need to understand what is going to be in these areas. So in these introns, we're going to have 3 conserved sequences that are always going to be present in an intron. And that's going to be that in our 5' end, so let's say this is our 5' end and this is our 3' end. At our 5' end, we're going to have a GU. Now, in case you guys are asking, what does GU mean? Come on, guys. Guanine and uracil. Understand that uracil is going to be replacing thymine in RNA. So thymine does not exist in RNA. Instead, it's uracil. So if I was to say, what is the complement nucleotide for adenine in RNA, it's uracil, not thymine. Just to get that clear. So here at the 5' end, we have this GU area. And at our 3' end, what are we going to have? We're going to have an AG area. And right in the middle, like I said, this is always present, we're going to have an A sequence. And this right here is going to be joining them. So right here, this is going to be joining them, and this is right here. So these are our 3 conserved sequences. So in case you guys forget it, let me write that down. I'm going to put right there. Our 3 conserved sequences in every intron. And we can know that this right here is an intron. Why? Because it is in the middle of the exons. X, you know, it's away. Let's say you break up with someone, it's your ex, get them away. Okay, so now that we understand that, we're going to have some small nuclear ribonucleoproteins. In other words, SNRNPs. Small nuclear ribonucleoproteins. And these guys are going to come in along with some other proteins to create what's known as a spliceosome. So it's going to come over here along with other proteins to create something called a spliceosome. So here I'm going to write spliceosome. Okay, so now this spliceosome is going to require several things, which is practically our SNRNP plus our pre-mRNA. So this is what required to create a spliceosome. Our spliceosome is going to have 5 SNRNPs, which will be known as our U1, U2, like the band, U4, so not U3, U3 is mean, so you got kicked out, U5, and U6. So these are 5 SNRNPs that are going to be necessary for us to create our spliceosome. And our pre-mRNA is going to contain, so in other words, what's going to help make this is going to be some snRNA and certain proteins. In other words, small nuclear ribonucleic acid and proteins. So now this spliceosome, now that it has been made, it's going to come and recognize that area. It's going to come and recognize that area. So right here, let's say our SNRNP. So this is technically, let's make that our SNRP. So we're going to go and put hard eraser so we can erase it. Go right there. And instead of putting SNRNPs, let's put spliceosome, our spliceosome. So our spliceosome is going to come and join this being our step 1. Let's separate it into 3 steps. Why not? So now that it has done that, what is it going to do? Well, if you guys can remember our different areas, our consensus areas, remember that I mentioned them? I really hope you did. I hope you guys were paying attention. What are going to be our areas that are going to be moved? So what we're going to do is we're going to move up a little. These areas right here that we had talked about, the conserved sequences, the spliceosome is going to kind of rearrange their movement to separate the exons or the intron from the exons. So to do that, we're going to have our GU, our A, and what color did I put it? I put it as green, and our AG. So these guys that were right in between our exons, if you notice, I kind of moved this a little. And that's because the spliceosome, which let's say I will put it here in kind of like a gold, is going to rearrange the unit into where this GU is not going to be connected to our exon over here. Instead, what's going to happen, let's say these used to be connected. It's going to create another connection with a little phosphate right here. Now this phosphate here is important because if I was to go up here, these connections right here all contained phosphates. So a little phosphate right here and an O, because well, technically we need to understand that they're an OH. And another P over here. Why? Because, well, we need to have these phosphates connecting. Okay, so now this is going to happen. And the phosphate that was over here on this with this exon is going to kind of create a certain movement in where now this exon is going to kind of try to go into this phosphate here with the OH end that it has. Why? Because if you guys could remember, this was our 5' end and this is our 3' end. So this area right here is the 3' end on this side, not that side. So here on this side of the exon is our 3' end that has the OH. And that OH is going to come and kind of create or bind to the phosphate that's right here. So now what's going to happen? Well, we're going to go into our step 3. And now our exons, so we're going to put this right here, are going to be connected. Why? Because they had just binded. Put our little phosphate there. Our exons are now connected. And we're going to have something known as a lariat intron. So here, now this has now been degraded. So it's going to go and become degraded after it has done its job. And what's going to end up? Well, now we have GU, our A, and our AG. Then we'll draw the little bonds right here. This one, and then this one. This one having the phosphate. And now this end, can anyone guess what the end has? Because it's not a phosphate. Here we're talking about a 3' end, so we have an OH. An OH part of the end. And this right here is going to be known as a lariat intron. And this now is our mRNA. Of course, this is going to happen in a bunch of exons. This is just a certain specific portion in which just 2 exons. But remember how we were talking about the previous one. So I'm going to go back to our previous one. We were talking about right here when it went through this process of going into mRNA and going in through some splicing. So if you notice, it happened with various introns, and this is how it occurred. In the creation of certain lariats that is going to help remove it. Now, one thing we also need to understand is that this is not the only form of splicing. We have very different versions because we also have what is known as an alternative splicing. And the only thing that this means is that sometimes, so here I'm going to go and put alternative. Sometimes, even though the exons are the ones that are transcribed, sometimes they're not the ones that we want. So here I'm going to put some 3 exons here. And if you want, I will actually add some color to them. So let's get rid of those. First exon being blue. Make this one orange. This one red. So these are our 3 exons here. And let's say now these are our introns that are connecting them. So our introns are right there in between. And let's say now that it's gone through our 3' cleavage and over here at this end over there, it's gone through that 3' cleavage and polyadenylation. I don't know if you guys remember when we talked about it. So let's say it went through that. And now instead of this being here, once it has gone through its 3', this is going to be gone. And what's going to be after our polyadenylation on this side? Lo and behold, we're going to have a bunch of adenines because this is our poly A tail. But now, through alternative splicing, we can have different forms in our lariat. Because one end that we can possibly have, so let's say I'm going to do this, because this is one way it can go, and another way it can go, is us having this. Or we could have this. So you're like, wow, wait, what happened to the orange? Well, you see, during the form when we were talking about splicing, maybe in the form when we had that lariat intron, so let's say I'm going to get our lariat intron, and it's going to look like this. Why? Because we have our union, here is our A, here's our GU, and here is our AG end. But now what could have occurred in this area here, that when we had this, not necessarily the ones that were right here, but a huge portion of it, in other words, the GU from this one combined with the A of this side. So now inside this lariat intron is this other exon that was kind of like removed. So this exon 2 is removed so we can have a different kind of mRNA. So that is another alternative way that we can actually just kind of consider. This is so you guys can understand that it's not always, well, this way that we have up here. But we can also go with an alternative in which certain exons are selected to be part of this highly exclusive mRNA. Okay, and that is everything you need to know about transcription in eukaryotes. In case you guys have any questions, any comments, be sure to send me a message. Maybe I got something wrong. Please be sure to tell me. I really hope this video helped you. Maybe if this is too long, maybe I can try to find a way to make it shorter. If you guys need anything clarified, please send me a message at techmed36@gmail.com. Thank you and have a nice day.